Your somatic cells have 2 of each chromosome. They are, what are they, haploid or diploid? Diploid. Somatic cells, germline cells, create individual gametes. Those are haploid. Gametes are haploid. What are these pairs of chromosomes called? They're identical, remember? Homologous. The specialized form of cell division that occurs in your germline is called what? Meiosis, right. Gametes that have 1 of each chromosome are haploid or diploid? Haploid. Yeah. Okay. You were on it. Okay. The genome. Well, it's not really an instruction manual, as you know. It has a whole bunch of genes with different alleles, but the phenotype that we actually have is an expression of the genome in terms of the environment that it's in. It's always that way. So, like when they talk about nature and nurture, that is an incomplete model. There's no such thing. Nature and nurture happen at the same time. Think about humans and language. You think nurture, language acquisition. We learn, right? That's nurture. No. Part of it's nurture, sure. You have to be exposed to language to develop language correctly. But without the substrate for learning language in our brain, we would never have language. It's both. Similarly, you have a heart that develops, right? You think, oh, that's just nature. The cellular environment, it actually starts out, a circulatory system actually starts out before you even have a heart. And there's actually a little bit of blood flow going on. It actually causes shear on certain parts of the blood vessel wall. In an area where the heart's going to develop, the shear actually affects gene regulation and upregulates and starts what's called an invagination. And then from there, the heart actually develops because genes being turned on and being turned off. In other words, the cellular environment is actually causing the heart to develop. It's not that you have a heart that automatically develops without those shear forces happening. Okay? So, when someone says it's nature or nurture, you tell them, no, you're wrong. It's both. In fact, it's explicitly both. You cannot have nature without nurture. You can't have genes all of a sudden being expressed or not being expressed without a corresponding environment, whether it's a cellular environment, the environment around us, the social environment. Okay? Doesn't work that way. Okay. This thing that I just showed you was kind of a misnomer. It was for a Bio 100 class, but I just want you to understand that a genome is not like a blueprint. It is not like that. Just like, actually, a house is not an exact expression of a blueprint. You've got different people building this house. They build an identical house, but not each house is actually identical, is it? In one case, you've got some guy who's a good carpenter, nails going straight. The other guy, oh, he bangs his thumb once in a while. They're slightly different. This group building the house, they're really a good team. The other group, you find out that things are a little off plumb. You're walking around and you're always kind of like going off to the side in one house versus the other. Okay. Now, hemoglobin gene. HbA, that's normal hemoglobin gene. Hemoglobin has an iron in the center it transports blood with oxygen. It actually transports oxygen, right? And for the normal gene, this you end up with a glue, I think it's a glucine or something like that, the amino acid. Okay? Now, occasionally, it turns out in Africa, an allele developed. It was a mutation. Mutations are sometimes good, mostly bad, or they don't have any effect. In this case, there was an amino acid change, and it went from GAG to GTG. And instead of coding for glucine, the amino acid, it coded for valine. Had an effect. HBS allele. Okay. And you can actually see what it looks like. So, you end up, especially when it's HBS, in other words, diploid, but the same allele, that means homozygous. For that, you end up with clumped hemoglobin protein. And that's why you actually get sickle cell anemia. Now, the interesting thing is, if you have an HBS and an HBA, you still get a little bit of sickling, but in the face of onslaught by malaria parasite, those individual cells, hemoglobin molecules that are caused by HBA, HBS, are actually difficult to get inside by the malarial parasite. So those individuals are actually resistant to malaria infection, and even if they do get it, they don't get it so bad. Unfortunately, if they have a brother or sister who's homozygous for HBS, HBS, those individuals have sickle cell anemia and have very, very, very low fitness. Okay. So in this case, this is called heterozygote advantage, or heterosis. But that's only in the case in an environment where malaria is prevalent. In an environment where there's no malaria, of course, HBS, HBS is always going to be bad, but it turns out oxygen metabolism is a little bit less efficient with an HBA, HBS individual compared to an HBA, HBA when there's no malaria. So actually, like in America, if you carry an HBS allele and an HBA, you don't have sickle cell anemia, but it turns out your oxygen metabolism is slightly a little less. And in fact, because of gene regulation and gene expression, some individuals who are heterozygous, HBA, HBS, can have significant sickling effect no matter what. So, this is what the gene looks like. It's all because of this one mutation. Now, yeah.
Are you able to, if there's a homozygous HBS, are you able to get malaria? You are, right?
Oh, if you're HBS, HBS?
Yeah, homozygous.
No, you can't get malaria at all. But the thing is, you're likely to die anyways. Okay? Now, it turns out that they're working on perhaps a cure for this with either gene therapy or something like that, where they can perhaps substitute an HBA gene into an individual and have it proliferate. In fact, well, there's a possibility, okay, but it's a ways off. There's also therapies that they can do to extend life, but still individuals who have sickle cell anemia, in other words, for HBS homozygote, they're fitness is usually pretty much reduced.
It's like a reverse mutation they're trying to do?
What?
Like a reverse mutation?
Well, it's kind of, it'll be like gene therapy,
Uh,
like we talked about with HMOs, not HMOs, GMOs and stuff like that. Or there's a new genetic editing technique called CRISPR that might allow us to change genomes that way. We'll see. Okay? Okay, so heterozygous, that means two different alleles. In this case, HBA, HBS. Homozygous, same allele. HBA, HBA, or HBS, HBS. HBA, HBA is normal wild type. Carry oxygen just fine. HBS, HBS, sickle cell anemia. Tibetans have an allele that's different, that actually enhances carrying oxygen. And it turns out it didn't arise, it only arose in the Himalayan, in the population in the Himalayas and the Tibetan plateau and spread. You can't find that mutation in, let's say, Native Americans in the Andes. Okay? It arose once. Similar to HBS, HBS, that arose, there was another mutation that actually arose in Southeast Asia. That didn't have quite as much problem in terms of sickling, but was also the same sort of protection against malaria. Okay? So it always depends on the environment, every time.